gradient potentials that we talked about last time. Okay, so let's review them, make sure we understand gradient potentials, and then we'll build on that and jump into those action potentials. Just for the wait for a second. All right, so when we talked about those gradient potentials, first off, let's talk about those ions and substances and talk about that resting membrane potential. What was our resting membrane potential? About -7 millivolts, right? And again, when I say -70 millivolts, keep in mind all I'm saying is the extracellular fluid, the fluid on the outside, has a different ion concentration and a different amount of substances outside than what's on the inside. So that if you could create some tiny little probe that you could stick down inside the neuron and then stick it in that extracellular fluid outside the neuron, you would see there was a -70 millivolts difference between the two sides of the plasma membrane. That's all I'm saying. Okay? So who was on the outside? Remember I said it was real easy to remember who was on the outside.
Sodium.
Sodium chloride. Table salt, right? Remember that? Sodium chloride. So we had sodiums out here. We had sodiums on the outside. Let's put some sodiums out here, and we had some chlorines. So just plain old table salt, right? All right? We good there? Who was on the inside?
Potassiums.
Potassiums. What kind of charge did they have?
Positive.
Okay. Hey, that doesn't help get -70.
Phosphate.
Right? Those guys don't help, do they? Because they're positive. We're trying to get to a -70, and they got a positive charge. That's not helping much, is it? Phosphates, PO4s?
Yep, negative. Protein?
Yep. PO4s. Okay, and then we said I'm going to use PR for proteins because most of your amino acids, proteins have this negative charge, so I'm going to put PR minus. All right, I'm not putting the leak channels in there, but the leak channels are important, aren't they?
Yes.
Who did we have the most leak channels of? Sodium or potassium?
Sodium.
Try the other one.
Potassium.
Potassium. There's lots of potassium leak channels. So potassium's constantly leaking out. Potassium leaking. Yeah, potassium leak channels would leak potassium. That's why they call them potassium leak channels. That's an astute observation. Can't argue with that. It's not what I was looking for. But yeah, potassium is leaking out, right? That is actually the most important as far as what's moving, okay? As far as creating our resting membrane potential. What about the sodium-potassium pump? Did he play a part of this somehow as well?
Yeah.
His job was actually to do what, though? Not so much to create the membrane potential, but to reestablish the resting membrane potential. Okay? That's pretty important. We're going to use him today. All right. So that's the distribution of the stuff. Okay? Inside, outside, and we create this -70 millivolt difference between the extracellular fluid and the intracellular fluid. Okay? Across that plasma membrane. All right, then we started moving stuff. Okay, and the first thing that we kind of opened was some of these sodium channels. We open a sodium channel, sodium's going to move
Out. Sodium is out. Inward.
Sodium's already out. So if we open up a sodium, let's say a chemically gated sodium channel, sodium's going to move in. If we move sodiums in, what's that going to do to our resting membrane potential? Make it more negative or less negative?
Less negative.
You guys agree with that?
If you're moving
Less negative. Positives and negatives.
If we open up sodium channels, voltage gated sodium channels, okay, or even I'm using voltage. We could use chemically gated. I think chemically is what we did last time, right? We were doing gradient potential. We open up sodium channels. Sodium's going to move in. And if sodiums move in, we're putting cations on the inside. What's that going to do to our resting membrane potential? Make it more negative or less negative? Less negative, right? Because there's fewer pluses on the outside, more pluses on the inside. And keep in mind, let's think about it this way. If I was to just grab that sodium right there and just completely pull him out of the picture, that would make the inside
It wouldn't affect it.
You don't think?
If you're just pulling it from.
If there's fewer sodiums outside.
That means it's less positive outside.
Okay.
So it could make it more negative inside.
You think that'd make it more negative on the inside?
I'm thinking.
I'm not.
Yeah, because it would have to balance out.
You said that the difference,
Yeah.
you said that the negative
Okay.
was the difference between the two. That's what you said.
It is.
So
Between
if
the
you
inside and
take
the
some-
outside.
So if you took something from the outside, logically, then it would have to affect the inside, because you
Okay.
said that negative
All right.
is the difference of the two,
Okay.
right?
If
Isn't that what you said? Am
Sure,
I right?
sure. If I when I move the sodium in, what happens?
It's less negative.
It becomes less negative. If I remove the sodium from the outside,
It'd be more negative.
No.
it's still less negative, isn't it? Because the pluses are on the outside.
But then the outside becomes more negative, doesn't it?
No.
The outside becomes more negative, so the difference
Oh,
becomes smaller, doesn't it?
You're right, you're right. Yes, you're right.
Okay? So when I here's my point. Here's what I'm leading up to. When I take these cations and move it to the inside, it's actually a double whammy, isn't it? Not only do I have fewer pluses on the outside, I have more pluses on the inside.
Mm-hmm.
So it makes a bigger change than if I just grabbed the sodium and removed it completely. See what I'm saying? Okay, so when you start moving these ions across the plasma membrane, keep in mind it's a double whammy. It's not just there's fewer outside. There's fewer outside, now there's more inside as well. So when it changes that membrane potential, it really does change that membrane potential. Okay? Hang with me, because when we get to action potentials, you can see this. Alright, so when we open up sodium channels, sodium comes in, the cell becomes less negative. We call that a what? Depolarization. Very good. We call that a depolarization, didn't we? Because it's polarized at -70, isn't it? Yes. And it becomes less negative, closer to 0. We are depolarizing it. Would you agree with that?
Yeah.
Okay. All right. What if we open up the potassium channel? Potassium would do what? It would go to the outside. Okay. Is it going to make it more negative or less negative inside the cell?
More negative.
It's going to become more negative. We call that a hyperpolarization. We could also accomplish a hyperpolarization by opening chlorine channels, and chlorine would move inside. And it would also become hyperpolarized. Everybody agree with that? All of those were graded potentials, correct? Say yes.
Yes.
Yes. When we talked about graded potentials, did they happen over the entire cell, or were they very localized?
Local.
Very localized. Did the signal stay the same? Did the signal stay the same? Write that down, just like I said it to you. Did the signal stay the same, or did it get weaker the farther from that point of initiation?
Weaker.
Weaker. Okay. So it's localized. It gets weaker the farther you go. And what kind of channels are we opening?
Chemically?
Chemically gated channels, correct? Does everybody agree with that? Yes, they are chemically gated channels. And again, if we open up sodium channels, we're calling the depolarization. We call that excitatory postsynaptic potential, right? Say yes.
Yes.
Yes. And if we opened up chlorine channels and or potassium channels, we call that inhibitory postsynaptic potential. So those were our two graded potentials. Is everybody up to speed? This is what we did last time. All this is review. We haven't started anything new yet. This is all talking about stuff we've already done.
So you were saying depolarization was excitatory?
Yes.
Okay.
Because, and we touched on this last time,
Yeah.
we were trying to get that -70 to what?
155?
We were trying to change it 15 millivolts, right, on the positive side, so we were trying to get it into 55 millivolt, minus 55 millivolt. And what part of the cell were we trying to get to that point? The axon hillock. That axon hillock right there, that cone-shaped region where the axon comes off the cell. That's the trigger point. We could change this part back over here on the back side of this cell And we could change it to -20. From -70 to -20. Is he going to fire? No, because it went in the axon hill. It's got to happen there in the trigger zone, or this cell's not going to fire. We're not going to create an action potential.
But you can have one that fires and fires and fires and fires
Yes.
on the other
Yes.
side.
And what did we call that?
Summation.
Summation. Very good. That's summation. When one cell fires rapidly, that was temporal summation. But what if a whole bunch of them fired all at the same time?
Another kind of summation.
Partial? Spatial.
Spatial summation. Remember that? Y'all looking at me funny like, no, I don't, but that's okay. All right. So again, here we were looking at a depolarization, right, by opening up chemically gated sodium channels. Okay, we're causing a depolarization. We're becoming less negative. So there's the -60. All right, so that would be excitatory or inhibitory. And now we're opening up, in this case, either potassium or chlorine channels. This would be a hyperpolarization, so this would be inhibitory, because we're moving it farther away from that -55 threshold, aren't we? It's going to make it even harder for this cell to get to 55 if we hyperpolarize it. Everybody with me? All right, we just, all we did was back up. We're taking a running start here to make sure we're all on the same page. All right, again, we looked at that right there. See, there's our little depolarization. See the little bump going to the up? Did we reach threshold in this case? No, we're not even close to it, are we? Okay, we just made one little bump right there, and that was it. We didn't get close to 55. So this cell didn't fire. Okay, so it goes right back down to resting membrane potential. And then in the other one here, we hyperpolarized. We became even more negative, didn't we? So again, we're even farther away from -55 when we hyperpolarize. All right, so let's jump to that slide right there and get started on action potential. And looks like our remote's out. So again, the happy place where we got to get this happening, we got to get to that little happy spot that we've been talking about, is in that axon hillock. When we create an action potential, notice that it is not localized, is it? Say no. No, it's not localized at all. When we crank it up right there at that axon hillock, that action potential is going to travel the entire length of the axon, all the way to the synaptic knobs. Okay? Very different than a graded potential, isn't it? It's not localized at all. Here we're going to open voltage-gated channels. We're opening voltage-gated channels in this case. How did we do graded potentials? Chemically gated, using neurotransmitters, right, to open up those gates. Now we're going to use that graded potential, that change in a graded potential, to cause the opening of the voltage gate. That creates your action potential. As we mentioned just a second ago, that threshold value is at 15 millivolt charge. We've got to change it typically from -70 to -55, and once we reach the happy little -55 in the axon hillock, then he creates an action potential.
And these don't require any energy at all to open. It's just a change in.
Right, we're going to use the energy to reset it, is what we're going to use, because that's where your sodium potassium pump comes in. Okay. Think of that membrane potential just like, you know, it's a potential. It's there. We've already created the energy required for it. We've taken the boulder and we've already rolled it up the hill, and it's sitting right there at the top of the hill. We used the sodium potassium pump to get that boulder up there, and now it's cocked and it's ready to go. Okay? What we're going to do here is we're not only going to depolarize, we're going to completely depolarize. we're not going to just slightly depolarize this thing. We're going to go all the way from a minus 70 to a plus 30, typically. So we're going to completely flip-flop the charge on either side of that plasma membrane, so that the inside becomes, for at least a brief moment in time, relatively positive compared to the extracellular fluid on the outside. We're going to drop back down to resting membrane potential, and actually we're going to overshoot the resting membrane potential. The cell actually becomes hyperpolarized for a slight moment. Here's another difference between action potentials and graded potentials. Action potentials keep their same intensity. We go from minus 70 to plus 30 right there in the axon hillock. Guess what we do at the synaptic knob? Minus 70 to plus 30. It's the same strength. Okay, again, we talked about in a graded potential, the signal gets weaker and weaker and weaker the farther you go from that point of initiation. Commonly, you'll see this phrase used when they talk about an action potential. It's all or none. When we reach minus 15 millivolt charge, and this thing fires, or if we don't reach minus 15, then we what? Right, we don't fire. That's exactly right. It's correct. Okay? If we reach threshold, we create an action potential. If we don't reach threshold, we don't create action potential. Right? Right. So again, it's all or none. It either fires or it doesn't fire. Graded potential was different, wasn't it? Because we could make a little iffy kind of thing. We could release some neurotransmitter, right? Or we could have a bunch of them release a whole bunch of neurotransmitters. Or we could have one of them get a whole bunch of shots, one right after the other, so that the neurotransmitter builds. Okay, nothing like this for an action potential. He either fires or he doesn't fire. All right. And the same intensity of response, no matter how much we depolarize. Now this is kind of interesting, because you would think, well, you know, if I change this right here from minus 70 to minus 55, okay, I get this action potential. Maybe if I change it from minus 70 to minus 30, maybe I'll make a stronger signal. Makes sense, huh? But it's wrong. I got it. Okay? But it'd be wrong. It's the same depolarization, repolarization, whether you barely reach minus 54 or whether you go to minus 20. It's the same signal that's going to go down that axon. You with me?
Can I ask something real quick?
Yeah.
So when they reach the minus 55, after it shoots it, does it drop down and you have to reach 55 again before it shoots
Before
the
it
signal
fires
again?
again, yes.
So
Yeah.
it's not
We'll
something
get to
that
that.
after, okay, so I got ahead.
Yeah.
Sorry.
Yeah. Don't get your knickers in knot. We're getting there.
And to get ahead.
All right. So when we create an action potential, flip-flopping charge, plus, minus, minus, plus, seems like we did all that. Where did we see all that before? Muscles! That's how a muscle cell created an action potential, wasn't it? Right? Didn't we see this exact same thing in a muscle? We opened up sodium channels and then we opened up potassium channels. Guess what we're going to do on a neuron? We're going to open up sodium channels, then we're going to open up potassium channels.
How about that?
It's the same action potential, isn't it? When you contract the head like this is yes. Okay. Sternocleidomastoid, this is no, right? Okay. All right. So let's look at the steps here. Here's where we're headed. We're going to depolarize the cell. We're then going to repolarize the cell. We're actually going to leave our potassium channels open slightly longer than we need to, so that the cell actually becomes even hyperpolarized from the minus 70. So hyperpolarized from minus 70 would be like what? Minus 8, minus 9. Okay, just making some numbers up here. Okay, so it becomes even more polarized than what it is. Because again, minus 70 is polarized already, isn't it? Because we're not at 0. If we were at 0, we would be unpolarized, completely depolarized. But we start off polarized because we're at -7. We're actually going to overshoot, and then we're going to come back up. And we're going to reestablish our RMP. What is that? Resting membrane potential. Man, you guys are sharp. All right, so let's talk about that depolarization. So when I say a positive change in the membrane potential, I don't mean just less negative, right? I mean, we're going to go all the way from -70 to a +30. Okay, so when we depolarize, I mean we really depolarize. So when that axon hillock reaches that happy little -55 point that we talked about last time, that's when we create an action potential. When enough sodium has come in, right there in the axon hillock, to depolarize the axon hillock from -70 to -55, that triggers the opening of these voltage-gated sodium channels.
Okay.
And when we, pardon?
I'm sorry.
That's okay.
I'm getting ahead again.
When we open up the sodium channels, what's sodium going to do? It's going to go in. Well, not out. It's going to go in, right? So we've gathered enough sodium coming in right here, into this axon hillock that it finally depolarizes enough. That's going to trigger the opening of voltage-gated sodium channels. And that's going to allow even more sodium to come in. Correct? Yes. So even more sodium is going to start coming to the inside.
So that's just going to take place in the axon?
In the axon hillock, that's correct. Because that's our trigger zone. That's our trigger zone.
So that's where the action potential occurs.
That's where it's initiated.
Initiated.
That's where it's initiated. It's in the axon hillock. So sodium rushes in, and the inside of the cell is going to start becoming positive. As more and more sodium comes in, what's that going to do to the sodium channels next to him that are also voltage gated? It's going to open in. And what's going to happen there? Sodium's going to rush in. So that part of the axon becomes positive. And what's that going to cause to happen on the cells, those voltage-gated channels next to them? It's going to cause it. Sodium rushes in, and we just completely repeat this process over and over and over and over, going in the direction of the synaptic knob. Now, you'll recall I ever so briefly mentioned when we talked about the sodium channels, I said there's really more than just open and closed. Normally it sits in the closed position, right? Say yes.
Yes.
Normally your voltage-gated channels sit in the closed position. All right. Well, the voltage change has caused these guys to open. Sodium rushes in. Well, when the sodium rushes in, there's a third state that this thing can go to. There's like another door that can close. Okay, we call that the inactive state. Why would we want to do that? Because we want to make sure the signal only goes in one direction. So it goes from closed to open to inactive.
Is it inactive by an enzyme, or does it just happen?
It's a shape change, because remember, these are proteins. These are proteins. It's just going through a shape change. Okay? So we're going to make sure the signal doesn't go backwards. It only goes in one direction. And that's because of those inactive states of the voltage-gated channels, sodium channels. Okay, again, I don't want to harp on this because I don't think it's a huge thing, but it is going to make a difference. And we'll pop a slide, bless you, we'll pop a slide up here in a little bit. I'll show you this is kind of a byproduct of that. Okay, but we got to make sure the signal only goes down the axon. You never want it to come back up. So it's got to go down the axon. Okay, so we open up sodium. Sodium rushes in. Again, only after we reach that little happy -55, right? Say uh-huh.
Yes.
Uh-huh. So now we're opening up voltage-gated channels. Sodium is rushing in, and now we're going not just to -55, we're going all the way to +30. And as that rushes in, it's going to open up the ones next to it. As sodium comes in there, that opens up the ones next to them. Sodium rushes in there, opens up the ones next to them. And we do that going down the length of the axon.
So it's very, very positive, really fast.
Yes, yes, but it's not going to stay there for long. okay? Because we are going to, almost immediately thereafter, we're going to open up, not chlorine, we're going to open up the potassium channels. And where's all the potassium right now? On the inside. All the potassium's on the inside. We're going to open up potassium channels. Potassium's going to rush out. Okay?
And this is all happening at the same time that the sodium's coming in, or it's happening after the sodium's coming in? Like, are they just...
It's immediately after the sodium.
Okay.
Sodium rushes in, potassium rushes out. I'll show you a little diagram in a second. A little graph that I think will help. Okay? So we depolarize by moving sodium, get to the plus 30. That opens up the voltage-gated potassium channels. Potassium leaves, and again, because the cations are leaving, we go right back down to the minus 70 again, don't we? We return to resting membrane potential. And it's just like we talked about for the muscles. Remember we talked about a depolarization and a repolarization by sodium moving out, potassium moving in. But we can't fire that cell again, can we? Because the ions are on the wrong side of the membrane. Sure, we're back to minus 70, but now our sodiums are on the inside and the potassiums are on the outside.
We don't like that.
And we don't like that, so we're going to have to fire up sodium potassium pump, put them back where they came from.
That's cool.
And then we can do it all over again. Okay? So again, it's just like the sodium. Just like the sodium channels, the opening of one set of sodium channels opens up the set of channels next to them. As sodium comes in there, it opens up the sodium channels next to them. Same thing's happening here. When you open up potassium channels and potassium leaves, that opens up the channels next to them. And potassium leaves there. That opens up the channels next to them. Potassium leaves there. So immediately following that sodium depolarization is this potassium repolarization. It just follows right along behind it. Okay? Doing that little flip-flop, that little diagram we looked at a second ago, that little flip-flop of the charges. Okay? So again, we do this just like we do the sodium, all the way down to the end of the synaptic knobs. Okay? So you see we start off again with this negative on the inside, positive on the outside. We flip that over when the sodiums move, and then we flip it back when the potassiums move. And that little flip-flop of plus to minus and minus to plus, we call that action potential. That's the action potential, isn't it? That's what you said, wasn't it?
Uh-huh.
That's what I thought you said.
It was.
It was. Okay. That's what we call an action potential. Okay? Just like in the muscle? Yep, we used the same word for it, didn't we? Same phrase. Okay. Now, those potassium channels are going to stay open ever so briefly longer than we need them to. So that a little bit extra potassium moves out than just to get to the minus 70. So we get to the minus 70, but because they stay open a little bit too long, and we're losing cations from the inside of the cell is going to become actually a little bit too negative, right? It's going to go to minus 70, but then keep right going. Correct? Say then minus 80. Okay? So that's a hyperpolarization. So the inside of that neuron very briefly becomes more negative, becomes hyperpolarized. And then we need to get everything back where they came from. So what are we going to fire up?
Sodium potassium.
Sodium potassium pump. Okay? We fire up the sodium potassium pump and we can reestablish our resting membrane potential. That's pretty groovy, huh? You feel groovy? Don't let me lose you guys. If you got questions, stop me. Let's make sure you understand what we're doing. So the graded potentials, again, through the process of summation, are going to get this cell either to the point of threshold or not. If we get it to the point of threshold, then the cell produces an action potential. If we don't reach threshold, he doesn't make an action potential. So again, when we talk about summation, keep in mind that he's listening to lots of other neurons that are talking to him, right? That are sending him signals. Some of them excitatory, some of them are inhibitory. But the sum of all those together is going to determine whether or not that axon hillock reaches that minus 55 or not. If he gets enough sodium channels open, despite the number of chlorine channels and potassium channels that have gotten open for inhibitory, if he can still get to that minus 55, boom, he sends a signal. Okay, and again, I always feel like I'm presenting this like that's what the neuron wants to do. You know, I mean, he's just dying to reach membrane threshold so that he can fire. You know, but keep in mind, a lot of times we don't want him to fire. I mean, that's not the point. You know, the signal needs to stop with him. You know, so keep in mind that everything's carefully timed and scheduled so that everything is sent like it's supposed to. Yes, ma'am.
So you're hyperpolarized and you've got a lot of potassium and you're positively charged. Is that right?
Not you talking about this hyperpolarization right here?
Yeah.
No, actually you lost a lot from the inside of the cell.
Okay.
Because remember these guys are positively charged, aren't they?
Yeah,
So,
that's what I'm trying to say from the inside of the cell.
yeah, but if we lose these from inside the cell,
Uh-huh.
that's what causes us to get hyperpolarized. Not
Okay.
having too many of them inside.
Okay.
It's by losing too many of them we become hyperpolarized.
Okay.
So when those channels stay open just a little bit too long, let me get to the curve. Okay, because I think this will help. I might even see if I can't get the pen to work on this thing, which is probably not going to happen, but we'll try. All right, so right here, all of this right here is summation, isn't it? Right? We're opening up what kind of channels right there? Chemically gated. Chemically gated. What kind of ion? Sodium. How do you know it's sodium?
Because we have to take the sodium.
Well, which way is it going?
Sodium's going inside.
Well, no. Which way is the curve going? Up. It's depolarizing, right? If we're depolarizing, what are we opening? Sodium channels, right? Because if we open chlorine channels or potassium channels, it goes down, right? So you said it right. Okay, we're opening up sodium channels. So there's summation right there. Okay? When we reach threshold, there's threshold right there. From right there to right there, what kind of channels are open? Voltage gated sodium channels. Voltage gated sodium channels. And the sodium's rushing in.
To make it more negative.
No.
That's what I keep thinking.
To make it less negative.
Well, yeah, okay.
No, there's a difference between more negative and less negative. Don't be,
Well,
oh
from
yeah.
70, negative 70 to negative 90.
No. No, we went to plus 30. Look at our graph. We're moving sodiums. What charge does sodiums have? Positive. It's a cation. We're moving them inside. So the inside is becoming less negative, becoming more positive, or less negative, however you want to say it. Does that make sense now? Okay. So that takes us all the way up to the peak right here. At the peak, who are we going to open up? Potassium channel. What kind? Voltage or chemical? Try the other one. Voltage. All this is voltage, right? Action potentials are voltage gated. Graded potentials are chemically gated. Okay? So we open him up all the way to right there because we stayed open just a little bit too longer than just getting to minus 70. Then we close those channels and we kick on the sodium potassium pump and it brings us back up, and now the ions are back where they're supposed to be.
-70.
Back at minus 70, so we're right back at resting membrane potential from right here on. We're back to resting membrane potential.
So sodium potassium.
I need to start over, or he needs to start over?
Well, I said you need to start over.
Yeah, and then he can do the whole thing over again. Yeah.
Sodium potassium happens at the -80. You see
Yeah,
that
yeah. You
right
keep
there?
it on down there at the bottom. Keep on sodium potassium pump. And reestablish your resting membrane.
So we start with chemically gated channels?
For graded potentials, yes, they are chemically gated. And what's the chemical? Neurotransmitters, right? And we're going to talk about them in this chapter before the end of it. But then the accumulation of all those different signals from those graded potentials, summation, if it manages to get the axon hill up to -55 because enough sodium channels have been opened,
That's the squiggly line.
those are the squiggly lines. Okay? When we get that axon hill lock all the way up there at that -55, boom, he sends a signal. He
And the
completely
voltage.
depolarized because we open voltage gated channels. So everything else is voltage gated from there, except the sodium potassium pump.
But then when you, yeah, you can write down it.
And then the repolarization's happening between 3 and 4 at the top, coming down.
Yes, yes,
That's
during
what we-
4, during
when
4
the
would be.
voltage potassium gated channels open, we're repolarizing.
That's correct, that's correct. You said that correctly. Does that help a little bit? Kind of see what's happening? Okay. Now, again, I don't want to spend a lot of time on this. There's actually some really cool experiments you can run if you got a physiograph machine and a spare frog to sacrifice. There's some really neat stuff that you can do with this.
You could come up with a frog.
But do I?
Find a frog.
We can find a frog? Okay. I don't know where a
Find a frog
physiograph.
at this point.
We used to do these a long time ago. Anyway, there's a brief period. And I'm not talking days here. I'm talking just, a very short period of time. When it doesn't matter what you do to this neuron, you could take a whole bucket of sodium and dump on top of him. You can kind of rip open the plasma membrane and just dump the sodium in, and he's not going to fire. Okay. And the reason for that is those sodium channels are in that inactive state. Remember I talked about they normally sit in the closed state. The voltage-gated channels, sodium channels, open whenever they reach the right stimulus because that -55 threshold is reached. And then they go to this inactive state. And again, it's just a shape change. And while they're in that shape change, you can't move sodiums back and forth. So you can't create another action potential until he resets himself back in that closed state again. And that's going to be after the sodium potassium pump has done all of his stuff.
And that's just it's just like a protection system for it to protect it
From the signal going which way?
the opposite, towards the axon.
Exactly. Up the axon. We want to make sure the signal doesn't. The reason we have this inactive state is to make sure the signal only goes one direction.
What
Okay.
happens if the signal go back?
Well, you can't.
So he can never do something with.
Well, he wouldn't be able to send it to anybody, would he? Because the only people he's talking to is at the end of his axon. He's not releasing neurotransmitters anyplace else. So if the signal goes backwards, it'd be like sending a motor command to your brain, and the brain would take his temporal lobes and shrug them and go, I don't know what to do with that. It's a muscle command. I don't have any muscle. So, he wouldn't be able to do anything with it.
But would it be destroyed if it
No,
did?
it wouldn't, it wouldn't implode or anything like that. It just wouldn't be able to do anything with it. So we make sure the signal goes one direction by having this inactive state. But the drawback with having an inactive state is there's a little period there of time when you can't send a signal. Okay, and that's the absolute refractory period. Are you with me on this? Okay, I don't want to beat this dead horse a lot of ways. I just kind of want you to be aware of it. Okay. This is really not a bad video, to be honest with you. It's okay. You'll notice that the sodium channels here have 2 flaps, and the potassiums only have 1. The only drawback really with this is that they're reading all of this to you, so it takes a very long time, and I don't have that kind of patience. So I typically click on it and drag it and kind of speed it up. So we open up sodium. Sodium's going to run in. Okay, sodium rushes in. We open up the potassium channels. Potassium rushes out. But then watch the little back gate on that sodium channel. Oops, wrong button. Okay, click. Now it's inactive. Okay, until it resets itself, it's in this inactive state with that little back door kind of closed on it. See what I'm saying? And that's going to make sure the signal only goes one direction.
Exactly.
Cool beans? Okay. That one's even cheesier, so I'm going to skip it. All right, so let's talk about what happens in the synaptic knob, even though you already know. This right here is definitely review. What kind of channels are we going to open at the end? Well, there's still voltage. They're still voltage gated. What kind of ion though? Calcium. Somebody's been getting ready for test on Thursday. We're going to open up calcium channels. Calcium's going to run inside. It's going to attach to the surface of the vesicle. It's going to move the vesicle over to the edge in a process called, try the other one. Exocytosis.
Yeah, there you go.
Exocytosis is going to occur, and we're going to release the material to the outside. So you've seen all this, right? Wow, lots of blank stares. That's not fun. Okay. Well, there's a lot of calcium on the outside.
Ooh,
Hey,
felt that.
hey.
Take that.
Go throw it back
Throw
at
it back.
So as that action potential comes down that axon, when it reaches the synaptic knob, it opens up voltage-gated calcium channels. Calcium rushes in. The calcium is going to attach to the vesicles. It's going to move the vesicle over to the edge of the plasma membrane. Exocytosis is going to occur, and we're going to release the neurotransmitter into the synaptic cleft, just like we did when we talked about muscles. Okay? We've done all this. This is old news. Remember last time we released it, it went across the synaptic cleft. It hit that little notch on the other side in that muscle. That little motor inplay on the other side, right? And opened up those chemically gated channels there. And what was the neurotransmitter we used there? Acetylcholine. Okay. Now we're just releasing different neurotransmitters. It could be acetylcholine, but it can be some 99 some odd others. Different types. All right. You've seen all this, right? This is old news, right? Okay. We've done all of that right there. This is how we initiated a muscle contraction, was that right there. Okay. All right. Releases into the cleft, moves over. It's the ligand. It opens up channels on the other side. If it opens up a sodium channel, then that means it's excitatory. So that's an excitatory postsynaptic potential. If it opens up a chlorine channel, it's an inhibitory. If it opens up a potassium channel, it's also inhibitory, because it's going to cause hyperpolarizations on the other side. If it opens up sodium channels, it's going to be depolarizing on the other side. That's what you were thinking. You didn't say it. What you were thinking. Right? So these are graded potentials again, aren't they? Yes. And if that cell on the other side gets enough graded potentials to get to minus 55, he creates an action potential. You talk to the next one. And on and on and on and on. Again, it's kind of slow mode, but that's just, wow, it'd be great if I had a pointer. Oh, there it is. Okay. So again, it's kind of slow motion, but there's the action potential, opens up those calcium channels, calcium rushes in, exocytosis occurs. There's your acetylcholine moving across. The synaptic cleft binds to those proteins, opens those channels up. And then in this case, it's moving in. Maybe sodium, so that would be excitatory. Okay. Hot dogs, hot dogs.
Diggity dog.
Hot diggity dogs. Okay, good. All right. Very briefly, let me talk about two factors that are going to affect how quickly the signal goes down the axon. First off is the diameter of the axon. The larger the diameter, the faster the signal. Okay. Simply put, the larger the diameter of the axon, the faster the signal is going to go down. The second factor, which is actually even more important, is whether or not it's myelinated. Myelinated axons can carry signals much faster. And as I mentioned when I talked about, what were the two cells? Neurolymphocytes, good. And which one cells are neurolymphocytes? Good. They were peripheral. Who was in the central nervous system? Started with an O, ended in lymphocytes. Oligodendrocytes. There you go, oligodendrocytes. Okay, those were the ones in the central nervous system. Looked like an octopus. Remember him? Okay. They were supplying the myelination in the central nervous system. I know you haven't reviewed that. That's okay. You got a test coming up first. As I mentioned, it's also going to be much more efficient because we're not going to have to require the same quantity of ATPs than if we do a continuous conduction, which is what we're going to talk about next. All right, still good? So let's look at the two different type of conductions here. And again, one of them is myelinated, one of them is not. Continuous is when you don't have any myelination. So we're going to have to open up sodium-potassium channels down the entire length. So that means a depolarization, repolarization every millimeter, right, of that axon. How long did we say these things were? Right? A couple of feet in length, maybe, for some of them. Some of them can be very lengthy. So you've got to do that depolarization, repolarization, and you have to do it all the way down that axon, every little millimeter, all the way down, opening up sodium, opening up potassium, okay? And then, of course, then you got to do what? You got to reset all the way one end to the other, okay, using one ATP to move 3 sodiums and 2 potassiums all the way down that axon, okay? Saltatory conduction. In saltatory conduction, this is where we have myelination present. And this is pretty cool because, again, we don't have to reset that membrane potential on those areas that are covered in myelin. Okay? So the action potentials only move along the nodes. You remember the nodes? What was the other name for these neurofibril nodes? The nodes of Ranvier. Right? So the action potential only has the nodes of Ranvier. Okay? Because the myelinated areas, now this makes sense if you think about it. Myelination, right? Myelin. Lipid? Yes, it's a lipid. Lipids are polar or non-polar? Ooh, that was a long time ago. Try the other one. They are non-polar, right? And that what makes a lipid is the fact they're non-polar. Hydrophobic. What about sodiums and potassiums? They're polar. They're not only polar, they're fully ionic, aren't they? They've got a full-blown charge. Do polars and non-polars like each other? Nope. So you take that axon and you wrap it up over and over and over and over and over with a whole bunch of layers of lipids. Man, them sodiums and potassiums, they can't do anything moving in or out, can they? See how it coats it? Keeps those ions from moving across those areas? Okay? Those nodes of Ranvier, they lack the myelin insulation. So lots and lots of voltage gated sodium and potassium channels. Huh? Oui? Oui. So how does this signal go, huh? How does this signal go down the axon? So, this signal, let me put it up. This signal jumps completely over, right? How does it do that? Well, opens up sodium. Sodium comes in. Sodium comes in, travels through the cytoplasm to the next node. Depolarize the next node. More sodium. Come in. Huh? Sodium goes through the cytoplasm. Next node. Sodium comes in. So you see the signal. The signal is just traveling through the cytoplasm until it gets to that next node. Opens up voltage gated channels there, sodium rushes in. That opens up the potassium channels, right? Potassium rushes out, okay? But the sodium that moves in travels through the cytoplasm to the next node. Opens up the voltage gated channels there. Now we only have to reset those areas where the nodes are, right? Because all those areas covered up in myelin, there was no ions moving back and forth there. See how it saves us energy? And the signal goes zip down through there. Instead of just having to depolarize and repolarize all the way down that axon from one end all the way down to the other. Cool? Cool. Any questions there? No. All righty.
So in the nodes, we're repolarizing or depolarizing?
Yes.
Repolarizing.
Yes.
In the nodes.
Yes, both.
Both?
You're doing the entire action potential action right there in the node.
Just in the node.
Just in the node.
Then
That's
what
correct.
happens in between the two again?
Sodium. The sodium that we brought in right here in the node travels through the cytoplasm to the next node, opening up voltage gated channels there.
Oh, okay, so it just shoots through it rather than voltage gated all the way
exactly.
through. I see it.
Sodium comes in, travels through, opens up voltage gated channels to the next
I
node.
gotcha.
And we just repeat that over and over and over again.
Thank you.
All right, you're welcome. All right, for neurotransmitters, there's actually a lot of these. There's like 100 of them known. And you probably don't want to learn all 100 of them. So let me just present to you the 4 classes, and you already know one class, acetylcholine, right? That's one class right there, so you're 25% of the way there already. Let me give them to you and then kind of give you an example, some of these that you've probably heard of that you might be more familiar with. Again, we'll start with acetylcholine because it's kind of the real easy one to think about. This past weekend I went to Fort Worth to the Anatomy Physiology Society meeting there, and this lady was giving a talk, and she didn't pronounce it acetylcholine. She called it acetylcholine.
What?
And she kept talking about acetylcholine. I kept going, what? I've never heard of that, And then finally she popped the term up, and okay. Apparently that's how they say it over in Texas over there. Acetylcholine. And I kept going, acetylcholine. What is acetylcholine? I don't know that one, But anyways, both CNS, PNS, and of course this is the one that we had in neuromuscular junction, wasn't it? So we know this one. We already know this one. Right? That opened it up to those voltage gated channels in that muscle cell. So we already know him. The rest of these you're going to notice are, if they're not amino acids, they're certainly related to proteins. So that kind of makes it a little easier. The second one here, amino acids, include one called glutamate, which is one of the amino acids. And I throw him out at you because this is actually the most common neurotransmitter of the brain. So obviously it is quite important. Again, it can be excitatory or inhibitory. It just depends upon what those receptors are on the other side, right? If it's a chlorine channel, It's inhibitory, right? It's a sodium channel. It's excitatory. Yeah, it just depends on what kind of receptors those are on the other side of that synapse. Monoamines are modified amino acids, some amino acids, not all of them, but it includes some that I know you've heard of. Adrenaline, as well as dopamine and serotonin, which again are very important neurotransmitters in the central nervous system. So again, I want you to stop and think about it. Acetylcholine, I can't pronounce it like that unless I stop and think about it. Acetylcholine, and then the second one was an amino acid. Here's a monoamine, and then the third one here is what's called a neuropeptide. Again, peptide ought to jump right out at you, right? Amino acids, right? Put a bunch of amino acids together, you make a peptide, okay? And that's going to include somatostatin, which is one of these CNS inhibitors. Okay, but then again, that just kind of gives you a cross section and some good examples of each. Still groovy? Still full of grooves? Okay. All right. Very briefly, again, not to get into a whole lot of detail here, but very briefly, there's two ways of getting rid of the neurotransmitter after it's been released. And I bet if I left you to yourselves, you could probably think of both of these. We can either break it down, right, degrade it, or we can pull it and reuptake it back into the neuron that released it, right? Say yes, because it's true, okay? So we can either degrade it, so we may release some chemical that breaks it down. So for example, acetylcholine is broken down by acetylcholinesterase, okay, ACHE. Okay, which is obviously just an enzyme that breaks down acetylcholine, acetylcholine. Or we can just completely reuptake that neurotransmitter, reabsorb it back into that neuron that released it. And if you think about it, that's kind of just, in fact, we could just stick it right back in the vesicle, couldn't we? Say yes. Yeah, we could completely recycle it, stick it right back in the vesicle, and store it in the vesicle until the next signal comes along, and then it's released again. The very exact same molecule is released again as a neurotransmitter.
Does that happen through endocytosis?
Yes, absolutely. Okay. Yep. Or it depends on the size of the transmitter, right? If it's small enough, yeah. Or excuse me, if it's large enough, yeah. If it's small enough, we might just use a transport protein and pump it back in using ATP. It just depends on the size of it. All right, nerves. Last little thing here to talk about in this chapter, nerves. Now, students love to use the term nerve and neuron interchangeably, but they're different critters, aren't they? A neuron is just one nerve cell, right? That either initiates or transmits a nerve impulse. Okay. A nerve is going to be a cord-like bundle of nerve processes held together by layers of connective tissue, because everything's held together by connective tissue. Absolutely. So here we're looking at a whole bunch of processes. Typically axons? Yeah, typically axons. And they're all just happen to be bundled up inside the same structure. Okay, that's a nerve. It's not an individual nerve cell. An individual nerve cell is a neuron. Here we're taking a lot of processes and we're just bundling them all up together. Good?
White stuff the dentist pulls out of your teeth. Inside of your tooth.
No. Where did that go?
The root canal when they pull the nerve out?
I don't know. Never had one. Okay, sure. I'm thinking more like frog legs and pulling the sciatic nerve out. Because if you don't, what does it do? Anybody try the frog leg without taking the...
Jump out of the
Yeah, it kicks. Kicks the grease all over the place. Yeah. Not real good thing to do on like a gas stove. All right, so obviously, structure-wise, we've got axons in there, right? Could they be dendrites? Yeah, they could be. But typically they're axons, because that's the long one, isn't it? Typically. Say yes. Yes. Are we CNS or PNS? PNS, right? Because there's only two parts of the central nervous system. Brain and spinal cord. Nerves and sensory organs were PNS. So there's nerves, right? These guys are bundled up into little sub-bundles called fascicles. I bet you've never seen that word before. You have?
Matter of fact, we have.
You have? Where have you seen that?
We just looked at it with the muscles.
In muscles. You're right. Muscles had fascicles. Fascicles was just kind of a little sub-bundle, wasn't it? Of skeletal muscle cells that was part of a muscle, just kind of a little sub-bundle in there. Fascicles is just a little sub-bundle of axons built into that nerve. Okay. I bet you guys haven't even learned those layers of connective tissue from muscle, have you? Surrounding each individual muscle cell would be... Shoot, I was joking. I was really sitting there thinking they know this, so this is going to be great. Now I'm scared because the test is in 48 hours, 47 hours, and you guys don't know it. Surrounding each individual muscle cell is a layer of connective tissue called endomysium. Endomysium. Surrounding the fascicle was perimysium. Surrounding the entire muscle was epimysium. That's scary. Okay. Well, there's connective tissue here as well. And the first one we're going to talk about is endoneurium. Endoneurium probably surrounds each individual... Not nerve! The whole thing's a nerve! Not the fascicle, that's peri.
Neuron.
Each neuron, each process, right? Each axon, or each dendrite. Yeah, I am scared now. You guys got a lot of work to do in the next 47 hours.
That's why they make coffee.
That's why they make coffee. There you go. And Red Bull.
Can't drink that.
All right. So surrounding each individual axon is this layer of connective tissue, okay, called endoneurium. Now, do you think you're going to put the Schwann cell outside of that or inside of that? Try the other. It's going to be on the inside because it's got to completely surround that axon, and then you put the endoneurium outside of that. Okay? Because you want that Schwann cell in direct contact with that axon. Okay? Then surrounding the fascicle is going to be perineurium. Good. Perineurium. It's going to wrap around the fascicle. And then surrounding the entire nerve is a layer of connective tissue called epineurium. There you go. Good. Now you're starting to see the pattern. Okay? Dense irregular CT surrounding the entire nerve. Are neurons going to require pretty good blood supply, keeping them up and running? Lots of glucose, oxygen, things like that? Say yes.
Yes.
Yes. So we should probably build blood vessels right into the nerve, shouldn't we?
Yes.
Yes, and we certainly do. We build them right there into it so that it's got a nice supply of blood because he needs glucose, he needs oxygen, he needs amino acids. Right? He's got to make neurotransmitters, doesn't he? And we said most of those were amino acids or protein related. So he needs a pretty good blood supply. Cool? Okay. Ooh, that's a good time for that to lock up, wasn't it? So there's a picture of it. I guess that's still catching it. All right, actually that's a great SEM up in the top corner right there showing you what it looks like. All right, last thing we kind of want to lead up here, we talk about classification and then what everything's hooked up to. First, structural classification. Obviously they're going to be hooked up to either brain or spinal cord, right? All nerves are eventually hooked up to either the brain or the spinal cord. If it's hooked up to the brain, we call it a cranial nerve. So if it's hooked up to the spinal cord, we call it a spinal nerve. There you go. There are 12 pairs of cranial nerves. They have fun names like glossopharyngeal and vagus and olfactory and vestibulocochlear, and we'll learn them in the next chapter.
Oh gosh, that brain-eating amoeba.
And then
It goes through olfactory and eats your brain.
well, see?
It's crazy.
It is crazy.
Okay, go ahead. I'm sorry.
31 pairs of spinal nerves, and we'll talk about them in the chapter after that.
Seriously?
We can also classify nerves based upon the type of information that they're carrying. Now again, keep in mind, guys, these are nerves. These are not neurons, correct?
Yes.
Two different critters. Neurons and nerves are two different critters. So when we classify them functionally, yes, we classify them as sensory nerves, but that just means they have a whole bunch of neuron axons coming from some sensory organ, correct? Yes. Carrying sensory information from sensory neurons. And then we could talk about motor nerves. And of course, it's not a motor neuron, although it will contain axons from motor neurons carrying motor commands out to effectors.
Is that efferent or efferent?
Efferent, outgoing. Okay? The third type, and again, this should really nail it home because there are no neurons that we would classify as mixed neurons, would we? Say no. No, there are no mixed neurons. There are mixed nerves, though. All we're saying is, here's this cord-like bundle of processes. Some of them, oh, this would work out good. Some of them are carrying, see where the cap is? Some of them are carrying information that way. Some of them are carrying information that way. We're bundling them all up together. So these guys are carrying motor commands this way. These guys are carrying sensory information that way. Those are mixed nerves. Okay? All of your spinal nerves are mixed nerves. They all contain both sensory and motor. All your spinal, yeah, we'll talk about that later. Can we use the same neurons to carry sensory and motor? No! We just talked about it. It can only go one direction down the axon, right?
One-way street.
It's a one-way street. You're either going to carry sensory, or you're going to carry motor, or you're an associational, so you're storing a memory or something like that.
We talked about association.
Guilt by association.
Again, most of your nerves are going to be mixed nerves. Again, you have 31 pairs of spinal nerves, and they're all mixed. So there's the vast majority right there. Okay. And again, as I just mentioned a second ago and panicked about because you gave me the wrong answer, right? Only one type of information. Can't carry both. Because you can't send different types of signals, can you?
No.
Right? Action potential is an action potential. Can this, let's say this neuron right here was a temperature receptor. We'll talk about this when we get to sensory organ. He's a temperature receptor. Can he send one signal saying, okay, I touched that and it's hot, and then you touch this over here and that's cold? He can't do that, can he? He can't send two types of signals, can he? An action potential is an action potential, period. Okay? He can't tell you that's hot, that's cold. So that means we're going to have separate receptors on it. Some for hot, some for cold. When you touch something warm, the hot receptors fire. When you touch something cold, the cold receptors fire. You with it? Because an action potential is an action potential.
I have a dumb question.
I bet it's not.
So when you touch something hot and it feels cold at first, are they both firing at the same time?
No, that just means your brain's right. Because to the rest of us, we touch something hot, it's hot.
But it's like hot and then it
Does that
cools
mean
down.
that the brain's
Yeah, I
wet?
had that happen.
Like a cup of
Yeah.
That's what I
coffee.
was talking about.
No, there's a cold.